thrilled to turn it over to the current medical director of the GW Center for Integrative Medicine, Dr. Mikhail Kogan.
Well, thank you, Lee. Thank you, Dr. Song, for making this happen and donating the seat to get this going. So I have an immense pleasure to introduce our first speaker today. Valter Longo is one of the world's leading experts on gerontology, and so I'm quite excited since my main field is geriatrics. Every time I get to hear him talk, it's always something new and exciting. He is the outmost expert on studying the fast mimicking diet, also assessing chronologic age, and not just chronologic age, but rather functioning age, particularly on cellular level and tissue level. He's Edna Jones Professor of Gerontology and Biological Science and Director of the Longevity Institute in the University of Southern California. His laboratory has identified some key genetic pathways to regulate aging. I'm sure he's going to talk about this today quite a bit. I understand that Valter is currently in Italy, so I hope that the technology will be our friend today. They'll be able to hear everything well, but that highlights the fact that he is traveling all the time. I'm sure he's going to talk about some of the Italian longevity. Of course, the part of Italy, Sardinia is in blue zones, so we're probably going to hear some of that about that. I am personally super excited, and I'm a little guilty. I haven't read his new book, Fasting Cancer: How Fasting and Nutritechnology are creating a revolution in cancer prevention and treatment. It just came out a few months ago, so I'm guilty as charged. I will be reading it soon. But I can't recommend enough his prior book that was in 2019, The Longevity Diet, one of the books that I give to a lot of my patients. Valter, thank you so much. We are so excited to have you with us, and we'll hand it to you.
Thank you, Dr. Kogan. Thank you for the invitation. And I have to say that I was asked to do a talk on cancer, right? So, so I'm not going to mention a lot of the things that you mentioned, but I'll focus on the cancer part. And so we've done maybe about half of my work is on cancer, so I like to focus on that. In this, I always show this slide as one of my initial slides, and this is actually the lung cancer part on the left is from a group in Switzerland, and the part on the right is our work, and this is showing cancer-free survival, right? So if you just treat mice with chemotherapy or fasting cycles, not much happens in terms of cancer-free survival. You get a little bit in a breast cancer model where we inject a few low level of cancer cells, but not much. And then you combine the two, and you combine the two, and now you see, at least in this model, it doesn't always happen like this, but in these models, chemotherapy, which doesn't cure much, now it's starting to be very effective in sort of searching and destroying cancer cells wherever they are, including in some metastatic models. So, so then, of course, we're very interested in the mechanisms on one side, but then using the mechanisms to actually come up with clinical trials that can test our hypothesis and the hypothesis that fasting and fasting mimicking diets can enhance cancer treatment. And so first of all, what are fasting mimicking diet? This started with a cancer clinical trial at Norris maybe 15 years ago, at Norris Cancer Center in Los Angeles. And the patients were placed on water-only fasting plus chemotherapy. And the patients did not want to do water-only fasting because it was just too much, too extreme. And the oncologists were not very comfortable with it. So from there we were funded by the NCI, the NIA, to develop a fasting mimicking diet. So it was developed for cancer patients. And so we've been almost exclusively both in mice and people, and in the clinical trial, we're using this fasting mimicking diet, which is a low calorie, low sugar, low protein, high fat, vegan composition. Okay, so then, so we've been publishing this for a long time, and one of the early studies, Cheng and Lee then in the lab showed that whereas in normal cells, AKT, TOR signaling, translation, these are all decreased during fasting. They're part of fasting responses, right? To turn down these signal transduction pathways and also protein synthesis. But in 41 and other cancers, in triple negative breast cancer and other cancer, we saw that in fact AKT, TOR, cis kinase was upregulated, right? So we kind of put it away, and then a few years ago we started working on this again, and started working on the idea that maybe there were escape pathways, right? So you starve cancer cells and normal cells are going the right direction. They know what to do. And this is a very ancient program, a fasting response program. But cancer cells are confused by this environment. Now you're reducing glucose, you're reducing growth factors, you're revolutionizing their environment. So they become desperate and they start increasing instead of decreasing pathway, right? So, and one of them was the TOR cis kinase pathway. So increase instead of decrease. So then Giulia Salvadori in the lab, and so I have two labs, one here in Milan, mostly focused on cancer. So Giulia then started looking at RNA-seq and basically looking at cancer cells and trying to figure out, this old observation that we made. Well, let's do it, looking at every pathway, right? So and so we fast the mice, we fast the mice with the cancer, and what happens in the cancer cells? How do they rewire, right? So do they do the same as we had shown a long time ago? And what are the pathways? And sure enough, Giulia then, by this RNA-seq, shows that PI3 kinase, AKT pathways, and TOR are in fact upregulated in these triple negative breast cancer cells. And so now, because TOR and PI3 kinase and AKT, now she targets those pathways with 3 different drugs in the hope that this would be enough to cause the regression of the tumor, right? So here's the tumor growth, the triple negative breast cancer. And now she intervenes with fasting plus the 3 drugs. And sure enough, right? So the cancer comes down. But the cancer then adapts and is able to grow, right? Unless there was a pre-treatment with the fasting mimicking diet. And so eventually we figured out why. And the reason was that if she pre-treated with the FMD and then treated with the 3 drugs, now the cancer comes down and stays down. And the reason for this is cancer stem cells, right? So then the cancer stem cells, so the fasting mimicking diet is narrowing this pathway is essential for cancer survival, right? And so it's narrowing this redundancy and is now making indifferentiated cancer cells, is making them rely on PI3 kinase, AKT, TOR, CDK4/6, and so a few pathways. But even though this is very effective with these differentiated cancer cells, it doesn't do much to the cancer stem cells. Now, the cancer stem cells have a different program. They're worried about sugar, right? These cells don't care about sugar. These cells are very dependent on sugar. And so now the fasting is lowering the sugar, and it's inactivating PKA, and it's also turning down these stemness genes that allow the cancer stem cells to proliferate. And so that's why we see, and actually in this paper, Julia actually demonstrated this, but this is why we see the cancer staying down, because the cancer stem cells eventually will give rise to new differentiated cells. And so the cancer keeps coming back unless in a glucose inhibition dependent manner, this problem is solved. Okay, so what if we started the other way, right? So what if we start with drugs that block in this case, hormone therapy. These are ER positive breast cancer cells. What if we start with fulvestrant blocking estrogen signaling, and palbociclib blocking cyclin, so CDK4/6 pathways, right? So if you do that in these tumors, it works just like in women that receive this for a while, and then the cancer, this green right here, the cancer starts adapting and eventually starts growing. And sure enough, in 1 or 2 years, in cancer patients, this particular standard of care dual intervention doesn't work anymore. But when Irene started combined it with the fasting mimicking diet, now you see that it keeps coming down, and then start seeing mice becoming cancer-free, right? So now, I mean, if you turn around, if we had started with the fasting mimicking diet, we will have concluded that CDK4/6 and estrogen receptor signaling was the escape pathway, right? So that's how we did it. We started with the standard of care and then used the FMD to block these escape pathways, to block additional pathways, escape pathways. And this sure enough works. And also it works in cancer cells that are already adapted and fast growing. So if we intervene now with the 2 drugs plus the FMD, now we see a reversal of the resistance acquisition, right? So then it's remarkable that Irene showed that so this is black and CDK4/6 is black, right? So the fasting mimicking diet was acting on 3 different things: leptin, insulin, and IGF-1. And if any of this was restored, the cancer will start growing again, right? So this is a really important point, because it says that if we did this for every patient, we will have to figure out all these different pathways that are important, because even between a cancer and a next generation cancer in the same patients, you could have different escape pathways. So now it would take 3 drugs to lower IGF-1, lower insulin, lower leptin, plus the 2 existing drugs. So it'd be 5 drugs required to get the same effect as we're getting now by adding a little bit. You can think about it as a wild card, right? So the FMD is making so many changes that then the 2 drugs alone are sufficient to do the job. Okay. So then Myra in the lab instead started thinking about, okay, so what is the problem with a lot of cancer therapy is the toxicity of the cancer. Even immunotherapy is very toxic, especially when you have multiple drugs being used. And so is it possible to do a very low toxicity or non-toxic therapy? And in this case, Myra started looking at vitamin C, right? So Luke Kentley, people had been testing vitamin C for decades, not very successful. And then Luke Kentley said, well, maybe it wasn't very successful because they were looking at the wrong type of cells. So he looked at KRAS and BRAF mutated cancers and showed that vitamin C treatment was in fact very toxic to these KRAS and BRAF mutated cancers. And so Myra in my lab confirmed this, right? So this is colorectal cancer, 3 different lines. So vitamin C is increasing the toxicity to these colorectal cancer cells. Doesn't do much to lung, more different lung cancer line, and a lot to the pancreatic cancer line. But now when she combines it with the fasting conditions, now you see clear synergistic effect, right? Now, again, like I showed you earlier between fasting and chemo, now we see it for fasting and vitamin C. And that's good news because actually both fasting, mimicking diet, and vitamin C are anti-aging interventions, right? So the opposite of what chemotherapy is, right? So they're both supposed to protect an individual. And so now the possibility is that you can begin to test non-toxic intervention to treat cancer versus the toxic ones. And so this is in vivo showing HCT 116 in vivo model. You see 3 cycles of the fasting mimicking diet are able to, together with vitamin C, are able to control tumor growth. They don't completely stop it, but they certainly have the biggest effect. And as we've seen for chemotherapy, the cycles of FMD and the cycle of vitamin C are about equivalent, right? But when you combine it, then now you see a much stronger effect. And so yeah, I don't have time to go through the entire mechanism, but essentially it seems to be working like this, that normally vitamin C, as Luke Kentley had shown before, is causing the generation of hydrogen peroxide. But this hydrogen peroxide, it needs iron, free iron, to perform phantom chemistry and generate a lot of hydroxyl radical. And the hydroxyl radical is what's really doing the damage to the DNA. It's that's where the toxicity is coming from. But heme oxygenase, which is and that's what we've shown. We've shown that in response to vitamin C, the cells activate this heme oxygenase, and heme oxygenase 1 is increasing ferritin levels, and the ferritin are basically scavenging iron, the free iron. So now they're removing this free iron availability, and so now the vitamin C is toxic, but not very toxic, right? So the fasting conditions now lower heme oxygenase 1, lower ferritin, make iron available, and now the phantom chemistry can go on, and it can go on specifically in the cancer cells, because as I'm going to show you in a second, heme oxygenase 1 is differentially regulated in normal and cancer cells. It goes up in normal cells in response to fasting, and it goes down in cancer cells. Okay. And so now we were seeing the fasting mimicking diets being effective in both immunocompetent and immunodeficient mouse model, and it didn't make many sense. And a lot of our colleagues were saying, how can it be, right? How can you see this working with or without the immune system? And so Stefano in the lab, the Biel's in the lab, started working on this and looked into both breast cancer and melanoma, and showing basically, this is the breast cancer model, that only in the combination of fasting mimicking diet cycles and chemotherapy do we see infiltration, T-cell infiltration into the tumor, right? So you don't see it with fasting alone, fasting cycles alone, and you don't see it with chemotherapy cycles alone, but you see it with both. And so this was true for breast cancer. It's also true for melanoma. And so connecting with what I just showed you for colorectal cancer, the heme oxygenase becomes a key player here too. And this is what I was saying earlier. During fasting in the liver and the muscle of mice, the heme oxygenase 1 will be elevated or greatly elevated. But in the tumor cells, now you see the opposite, right? So the fasting conditions are decreasing heme oxygenase. So in this case, it was enough during fasting to increase heme oxygenase to then restore the ability of the tumor to grow. And okay, so then of course the interesting thing was, well, what about if you combine it with immunotherapy? And so we had been working for a while on this, and then we got scooped by a group in Spain, and they combined it for lung cancer models, different lung cancer models, and they were able to show that in fact fasting conditions plus PD-1 checkpoint blockade was able to add very strong effects against lung cancer models. So Salvatore in my lab more recently then decided to combine PD-L1 inhibition with OX40, and OX40 is an activator of T-cell dependent tumor attack. So PD-L1 removing the checkpoint, and then increasing or activating OX40, and therefore pushing the T cells to attack the tumor. So the two are being shown to be more powerful together than each one alone. And so interestingly, the immunotherapy, and this we've seen in many models that we use, the immunotherapy does very little to the cancer progression. This is again 41 triple negative breast cancer. What works is either FMD plus OX40 or FMD plus OX40 plus PD-L1 inhibitors. And so why does it work? Well, Salvatore did single-cell RNA-seq, and in fact showed that compared to the immunotherapy, to the two immunotherapy drugs alone, which is here in lane 2, the FMD plus the immunotherapy drugs cause a major activation in at least 3 different populations: activated CD8 cells, TCF7 positive T cells, and CD4 T cells, right? So these groups are undergoing a major expansion. And Salvatore also showed at the single-cell RNA-seq level that, in fact, the number of activated CD8 cells was flat until the FMD and the 2 checkpoint inhibitors were combined. And this is just showing the expression, the gene expression typical of activated T cells. So this is showing that going to a different triple negative breast cancer model, showing that this is not about 41s, and this is so Salvatore used this TSA, and again now working even better. Now the combination of FMD, OX40, and PD-L1 is flattening the cancer growth. Okay, so another thing that I mentioned earlier is the toxicity, right, of immunotherapy, chemotherapy, and lots of other therapies. So interestingly, we started seeing in the 41 model, when we use either PD-L1 inhibitors, OX40 agonist, or PD-L1 OX40, all the mice died, right? So and what happened is this major activation. This is OX40 PD-1. You see major activation of eosinophils, major activation of basophils, or not activation, but increase in number of basophils, and immature granulocytes. And in all cases, you see that if you add the FMD back down to normal in all 3 different populations, right? So this may explain why the FMD plus the checkpoint inhibitor now is protecting, not only is causing a much improved killing of the cancer cells, but it's now preventing this very clear death of the toxicity of the therapy, probably caused by this activation. Okay, so then the last model I wanted to show you is a blood model. This is acute lymphoblastic leukemia, ALL. And Roberta in the lab look at the standard vincristine treatment, so chemotherapy alone, fasting mimicking diet cycles alone, chemotherapy alone, or the 2, right? So the 2 combined, again, not only improve survival, but long-term survival, like cancer-free survival, right? So now get about 60% of these ALL mice to be cancer-free long-term. And so why is this? Well, Roberta showed that the fasting mimicking diet conditions are now inhibiting lots of autophagy markers, particularly in combination with vincristine, right? So now the autophagy markers are down, and this autophagy is probably at the center of this increase in apoptosis, again, only in the combination of fasting plus the chemotherapy. And so then Roberta looked at what if I just use an inhibitor of autophagy and use chloroquine, and sure enough, the combination of chloroquine plus the vincristine, so chemotherapy plus autophagy inhibitor, was sufficient to get this cancer-free survival, long-term cancer-free survival. And again, she showed that if we use an anti-CD8, so if we remove immune T cell function, that effect goes away, and so we are now the cancer starts growing again, and it reverses the effect of chemo plus vincristine. Chemo plus FMD on cancer progression. Okay, so this is just the summary of that. So normally fasting causes autophagy, increased autophagy. Here again, it turns it around, right? So now fasting is decreasing autophagy, and this decrease in autophagy together with the chemotherapy is essential to promote cell death, and it promotes cell death in a immune-dependent manner. Now, probably we're still looking at the mechanism. Why are the two things working either with or without the T cell presence? And probably, although it works better with the T cell presence, probably because a lot of these mechanisms are free radical dependent. So hydroxyl radical, superoxide, hydrogen peroxide. So then the damage is done in the cancer cell by the free radical, but this is probably also making it much easier for the T cells to recognize the cancer cell, right? So this is our hypothesis that is a cell autonomous damage attracting immune infiltration. Okay, so and this is just the whole thing sort of put together. Where the fasting mimicking diet is really going after lots of different systems. So it's lowering IGF-1, it's lowering glucose, it's lowering insulin, it's forcing the cells to go back to a mitochondrial-dependent oxidative phosphorylation, even when the cancer cell can no longer use mitochondria because mitochondria may have been damaged, or the genes that are involved in mitochondrial respiration are damaged. And so and often we see this burst of reactive oxygen species, and now this ROS can do damage to the nucleus and to the cell in general. And again, now we think that all this intracellular damage now is making it potentially is affecting T regulatory cells, and that's also something we see, a reduction in Treg. But either in a Treg dependent manner or Treg independent manner is allowing the T cells, the cytotoxic T cells, to attack and continue the destruction of cancer cells. Now, there's now also a very interesting macrophage NK cell story. Now, this immune world is very complicated. So now we're looking at every cancer, every immune cell type, to see what else is involved and why. Okay, so what about clinical trials? Well, there's maybe 20 trials now that have been completed with fasting, mimicking diet, or fasting and cancer. One of the largest one was this one done in Leiden, 125 patient, HER2 negative, stage 2-3 breast cancer. And so in this trial, the group at Leiden saw a major increase in the non-responsive population. If we look at imaging, so clinical response, the regular diet, the patients on a regular diet were much more likely to not respond to the chemotherapy. And these were chemotherapy cycles given before the tumor were removed from the patient. And now, if you break it down based on the ones that were compliant to the receiving the fasting mimicking diet cycles versus the one that were compliant to the regular diet, the control diet, now you see a 5-fold difference in the portion non-responding. Now this is we just looked at the data. It doesn't look like this is very impressive, and I'll show you in a second how this will be confirmed with triple negative breast cancer metastatic. But this is probably not the stage where FMD is going to be most effective. Why? Because here's before surgery, the patient just do a few cycles of the fasting mimicking diet, and then that's it until, you know, the cancer may progress and patients go on to do something completely different, right? So this was showing fundamentally what we see in mice is happening here, but again, it doesn't look like this is probably going to be just a few cycles of FMD are not going to make that big of a difference in overall survival. Okay, but the Vernieri group instead is looked at lots of cancers as well as the Nencioni group in Genova. And in this paper published a few years ago, they saw evidence for the fasting mimicking diet cycles reshaping systemic and intratumoral immunity, right? So this is good news, and it's probably the same infiltration that we showed for the mouse studies they saw in patients, and this seems to be very consistent. So that's good news, suggesting that maybe that free radical dependent damage that we saw in the mouse cells in lots of different mouse models, now they're also occurring in people, and the T cells recognize that damage and attack it. I mean, I'm speculating here, but certainly, so the evidence is there. Now, whether that's a mechanism or not, we don't know yet. Okay, another interesting thing that they published recently was out of that trial that I just showed you, they had, I think, about 20 stage 4 metastatic cases. And out of the 20, 5 of them showed what they call exceptional tumor responses, right? So stage 4 metastatic type of cancers that you don't usually see going into remission. But here, these 5 cases, I think it was colorectal, breast, pancreatic, and lung. And all of them went into remission in a very surprising way. So again, you know, anecdotal, sure, but very interesting. And again, matching the still preliminary but long-term data. Here is triple negative breast cancer. So this is 41, and the triple negative breast cancer either treated with chemotherapy alone or chemotherapy plus the fasting mimicking diet cycles. So now this is a much more promising way to use the FMD long-term together with the chemotherapy, not just a few cycles, let's say very early on before surgery, and then, you know, hoping that this is going to do wonders, 5 years later. So yeah, so now we're seeing Ligorio and Vernieri saw a nearly doubling of overall survival after almost 4 years. So some of the concerns have had to do with muscle loss. And so here in Genova, they looked at the 8 cycles of the fasting mimicking diet and showing that in patients, in women that were doing this, there was no decrease in hand grip strength. In fact, if anything, there was a potential increase in muscle function and a potential increase in lean body mass, not decrease, but increase. And then this is very consistent with what we see in the diabetes trials, in the pre-diabetes trials, and lots of all the trials that we ran. The fasting mimicking diet has a short-term effect on muscle mass, but then long-term, the absolute mass stayed the same. The relative mass, lean body mass, actually increases. Okay, and then this was just published by the Vernieri group, and showing again, well, actually mechanistically showing the early down modulation of tumor glycolysis was predicting whether the triple negative breast cancer patients were going to be sensitive to the fasting mimicking diet or not. But the really nice thing is that, and this was FMD plus chemo plus or minus metformin, right? And the metformin made no difference. So all the major job was done by the FMD, and this nearly doubled or had a major increase in the complete pathological response, and nearly doubled the complete pathological response. So this is a surrogate for overall survival. And so matching the other papers that the Vernieri group had published. Okay, so I'm just going to end there. And since I cannot join you later, if anybody has questions, you can please ask me. Yeah. Thank you.
Great. Thank you so much. We definitely do have some questions. I think that a little refresher on what makes the fasting mimicking diet unique in terms of its formulation would be helpful. And how does that compare to, say, fasting? How is it different than fasting in terms of, what you are eating or are not eating?
Yeah, I always say that fasting really doesn't mean anything. It's like saying eating, right? Well, is eating good for cancer? Well, it depends
Right.
on, lots of details, right? Yeah. So I mean, the fasting making diet is a way to try to bring the standardization. I mean, first get the effects of fasting, and then standardize it, and then protect the patient from hypoglycemia, from the hypotension, and lots of problems that can occur. I mean, most physicians will say don't do fasting at home, not long-term fasting, and so the fasting mean diet is standardized that and made it so that people can do it at home. Now, in formal trials, it's been tested probably on an over, 1,000 cancer patients. So, so far we really don't see a problem with it. And yeah, so I think again, that's how it got started, right? It got started with water-only fasting and patients refusing water-only fasting. And then, we moved to the FMD. Well, we moved to developing the FMD and then testing only the FMD. And now all the hospitals that come to us for, so I think there are now 20 trials or more ongoing. And I think it would be very difficult to get these hospitals to do water-only fasting trials, right? But they're all excited about the FMD type of trial because, it comes in a box, it's given to the patient, and it's pretty straightforward for the patient. And the patients, in most trials, there have been there has been high compliance.
Mhm. I like to think of the fasting mimicking diet as a diet that both feeds your gut microbiome, your gut microbes, with fiber, and then also feeds your brain with healthy fats. That's my impression of it. I think there's a little bit of confusion as to, can you do it? Does it have to be vegan? Can you do this with animal protein? One of the questions in the chat.
I don't think so because of amino acid composition, right? So we're really targeting lots of the amino acids that are very low in the vegan in the vegan proteins, so methionine, leucine, et cetera, et cetera. Yeah, so I would say, and sometimes the difference is over fivefold, right? So this is not a 10% difference between an animal protein and, let's say, a legume protein. It's over fivefold. And so you can see how now, if you increase the level of an amino acid by fivefold, this could, interfere with everything we're showing you.
I have a question for myself. One of the things I've been thinking out, and I haven't seen it specifically studied, but I feel like you probably know the answer to this. One of the things that is more popular in athletes and high-end users of supplements is the use of amino acid supplements. And how can amino acid supplements affect this pathway? Obviously, it's not what you're typically studying, but right? Like you're talking about methionine is something that's really going to activate mTOR, and that is going to potentially undo some of these benefits.
Yeah, no, we studied the amino acids, right? So we're very, in fact, we were about to publish several papers on amino acid. And so, yeah, for cancer, this is particularly problematic, right? Because lots of cancers will survive if you give it the right amino acid, right? And so they thrive on, let's say, glutamine, and there is a few amino acids that the cancers love, and love in a metabolic sense, meaning they can be a precursor of lots of metabolites that they need to stay alive, right? And yeah, so then for a cancer patient, it's a bad idea. For a normal person, it depends, right? If you're malnourished, it's very good, right? If you don't have that amino acid, then it's starting to affect your immune function, your muscle mass, etc. Yeah, then you need it. Most people are not malnourished. Most people have too much proteins. And, and so these amino acids are at the center of increasing growth factors and increasing anti-apoptotic factors. And so, yeah, so this is why the mice that, and the people, but the mice that have the record longevity extension have a mutation either in the growth hormone or in the growth hormone receptor. And so this is the central amino acid pathway, if you want to call it that, right? So blocking the amino acid response pathway is causing this 40% lifespan increase. And then interestingly, if you take the mouse and you calorie restrict it, you further cut the amino acids in the diet, then they go to, this is work by Andrew Barkey, they now live twice as long, which is really remarkable, right? To make a mouse live twice as long.
I think Dr. Kogan,
Yeah,
I know
so,
Dr. Kogan's going to have questions.
well, yeah, so my question is obviously going to be a bit more clinical. So, Walter, I mean, I think it sounds like we getting close to say there is enough high quality evidence to say maybe it's time to start talking to the patients about implementing this on a broader scheme, especially since we know the safety here is really good. In the worst case scenario, well, we may not be necessarily benefiting every cancer, but we definitely going to be safe. And in clinical realm, people seeing your work and weird patients asking, should I be doing this? And of course, you can't really answer every clinical situation, but I want to get your general take on things.
Yeah, I would say for diabetes, pre-diabetes, this is much more advanced, right? Much simpler. So we now have done 5 trials and there's more coming and very consistent effects, right? So I will argue that there is a competition for GLP-1 that is much safer, much more going after the problem and not putting a Band-Aid on the problem, like much more real treatment than management, right? So now, a lot of these drugs are managing the disease. For cancer, I think that it's more complicated, and and it depends. It's 100 cancers, 1,000 therapies. So I think for sure, what we usually say to oncologists is, if you're running out or you're about to run out of options, then talk to us, right? Because now we can strategize and we can say, oh, this is not like, I just presented, let's say, hormone therapy. Well, it works, and then the patient becomes resistant. Let's say now the patient became resistant with fulvestrant and palbociclib or ribociclib. Okay, now talk to us, right? Because you may be in trouble, right? So what do you do next? Well, okay, you could try things, but yeah, so there's not too many viable options. So at that moment, I think it's good to start thinking about the fasting mimicking diet. In early cases, like I showed earlier, stage 1, undergoing surgery, 96% survival, I would say let's wait, right? Because what if this is making things worse, for example, right? I don't know, but it's certainly always possible.
Well, I think that's very exciting. I know it's been a while since we've had some major breakthroughs in cancer. We got the immunotherapies, which are fantastic, but they only work for certain cancers at this point. And it seems like perhaps they could work even better if we could put them with this adjuvant therapy, which has pretty low risk. So I'm personally very excited about this. I hope that this will be something that in the future we'll be able to know exactly who's going to benefit from this and start using it with whatever the best combination therapy is from the get-go and have better outcomes.